Okay, so welcome to this European webinar series on Microglia. I'm Guy Brown from the University of Cambridge, and my co-host today is Jake Dundee. And the series consists of monthly talks for 45 minutes by European scientists talking about their research on microglia, followed by a Q&A session. So today's talk is by Christian Haass from Munich, and the talk is entitled Microglial function and dysfunctions of TREM2 and ABI3, two major risk factors for late onset Alzheimer's disease. During the talk, please write your questions in the chat function and the Zoom at the bottom, and address your questions to all of the speakers. Please keep your microphones muted and cameras off to preserve bandwidth. And I'll now hand over to my co-host Jake, who will introduce the speaker. Jake.
Thank you very much, Guy. So here today we've got Christian Haass, who attained his PhD at the Ruprecht-Karl University in Heidelberg, and then followed that up with a postdoctoral fellow in the lab of Professor Selke at the Center of Neurological Diseases in Harvard Medical School. Christian then proceeded to become an assistant professor of neurology at Harvard Medical School and to have moved on to professor of molecular biology at the Center Institute of Mental Health at the University of Heidelberg in Mannheim. Currently, Christian is a professor of biochemistry and is the head of the department of the metabolic biochemistry at the Adolf Butenich Institute, as well as being a speaker for the German Center for Neurodegenerative Disorders. He's received many awards, including the Hartwig-Piepenbrock Prize for research into neurodegeneration, as well as the Brain Prize. Now, Christian's going to give his a talk on research surrounding microglial function and dysfunction in relation to TREM2 and ABI3, two major risk factors for late onset Alzheimer's disease. And with that, feel free to take it away, Christian.
Okay, thank you very much. So I hope my... somehow I can't proceed. Oh, no, it works. Okay. So thank you very much for the nice introduction. Thank you for inviting me to talk. So what I want to do here is I want to basically show you that the amyloid cascade itself is, to my opinion, still correct. It may not be as linear as it's shown here, but I want to convince you that microglia are an integral part of the amyloid cascade. Now, the whole thing started out, and actually I have to thank here Sir John, because he talked me into this business a long time ago when he found TREM2 being a major risk factor for late onset Alzheimer's disease. And of course, we then found that TREM2 undergoes proteolysis, very similar like APP, so it's cleaved by alpha secretase ADAM10 and ADAM17 to generate a soluble TREM2 and a C-terminal fragment. And then we looked at the first mutations, which by the time were really strong mutations, actually. And they misfolded TREM2, so it was retained in the endoplasmic reticulum. There was no surface transport anymore. Therefore, there's no cleavage because the secretase, alpha secretase, cleaves only on the cell surface. So there was very little soluble TREM2 and almost no C-terminal fragment. And that has functional consequences. So the microglial function really went down. The phagocytic function really went down. And that really then brought me really strongly into the field. And by now, basically, the entire lab is working on microglia-associated risk factors. And that's what I'm going to talk about today. We'll give you a short introduction and then show you also a lot of unpublished and brand new findings. Now, this was a result which really opened my eyes. So this is a TSPO imaging of just regular wild-type mice, which everybody of us has in the lab. And during aging from 2.5 months to 12 months, these mice are activating the microglia, as shown here by the reddish color of the TSPO imaging. So during normal aging, microglia become activated. And they do so because there's minor myelin damage. And that's what they respond to. So a normal microglial response, the activation, cannot be something bad. It's most likely a defense response. And that became very clear when we knocked in a TREM2 mutation with the CRISPR technology, just the T66M mutation, so only one codon was changed. And that basically, starting from 8 months on, stopped microglial activation. So these microglia cells, they were locked in a what we call homeostatic state, and that's apparently disease causing. So obviously we need microglia which can respond to a challenge. And that response, to at least to some extent, is protective. And I will show you later on that we can even pharmaceutically manipulate that response. Now, by that time, we also made a very surprising observation, which I couldn't really interpret at the very beginning. So we looked at FDG PET, and that's all still work from Gernot Kleinberger, who was a postdoc at the time in my lab, together with Matthias Brendel, who's running all of our PET images here in Haass, and who I really have to thank for his fantastic work here. So what we found here was really interesting. So the T66M mutation reduces the FDG signal. And this is just a single point mutation in a gene which in the brain is expressed only in microglia cells. So how do we interpret that? Does that mean that the signal, basically the FDG PET signal, is basically coming from microglia cells? And we asked the question by the time in the discussion of the paper, and now very recently, Xian, a very, very gifted PhD student who's now back in China working in her own research group now together with Matthias Brendel, then investigated that phenomenon again. And what we did here is we looked at APP-PS1 mice. And these mice have a hypermetabolism. And when we cross them to a TREM2 knockout, the FDG PET signal dramatically drops. And again, we also confirmed what we found before. If you have a wild type mouse crossed to a TREM2 knockout, the signal also drops once again. And then we went directly to microglia and thought, let's wipe out microglia. And of course, you can do that with plexin. And if you reduce the microglia, number of microglia dramatically, actually, with plexin, the signal drops once again. And similarly, even in the wild type animals treated with plexin, the FDG PET signal drops. So we interpret these results that in a way that microglia contribute substantially to the FDG PET signal in these mice. And not only that, Xian then developed a technology which Matthias then called radio sorting, a term which I like a lot. So the experiment is in principle very simple. So you take a radioactively labeled FDG PET tracer, inject it into your mouse, and then you sort microglia, astrocytes, and neurons. And then all you do is count the radioactivity. Very simple experiment at the very end. Xian did it in 3 different animal models, in wild type animals, in an amyloid model, and in the TREM2 knockout. Now let's first look in an amyloid model. In that case, we used APP knock-in mice. And what you see in blue, we have microglia. In red, we have astrocytes. In yellow, we have neurons. And you see that specifically in the microglia, the FDG PET signal in the APP knock-in mice was drastically increased as compared to the wild type mice. And when we look in the TREM2 knockout, it was again the microglia population where the signal for the radioactive tracer was selectively reduced, and not the astrocytes or the neurons. So that in fact speaks for microglia contributing directly to the FDG PET signal. Of course, neurons have their share, and we may lose synapses, for example, during the isolation procedure. So the neurons may be underrepresented. I totally admit that. But nevertheless, the microglia, they have a substantial portion and a substantial contribution to the loss of, or to the FDG PET signal. Now, of course, nowadays, we all know that microglia switch from a homeostatic state to a disease-associated state. I think we should discuss the names. I'm not very happy with that because disease-associated sounds bad and homeostatic sounds good. And I think this is not necessarily correct, as you will see throughout my talk. And nowadays, it's even clear that if you have a TREM2 loss of function, the microglia is locked in a homeostatic state, which already shows you that this is not necessarily a good state. So microglia need to be able to respond to a challenge. And if they cannot do that, you have very obvious phenotypes. So normally microglia nicely accumulate around amyloid plaques. These in green you see an EBA stain, and in white you see amyloid plaques, and basically all the microglia are located now at the amyloid plaques. Now, when you look at the TREM2 knockout, this is not the case anymore. And then by the time Samira and Thomas, who runs our neuropathology department, or helps us with the neuropathology here at our department, also looked at TREM2 patients. So we had a total of 7 TREM2 carriers with Alzheimer's disease, and I'll show you 3 examples here. And all 3 examples, as compared to the regular Alzheimer's disease control, in that case, had less microglia sitting around or on top of the amyloid plaques as compared to the regular Alzheimer's disease case. So this was, by the time, quite a surprising result for me, because the mutation, of course, in humans is heterozygous, and what we are talking about here is a full knockout. And that makes it, of course, a big difference, and I didn't expect such a significant difference to see in heterozygous patients. But indeed, that was the fact, and that shows you how potent a TREM2 loss of function is even in humans. Now, beside TREM2, there's another, not a risk factor in that case, a factor where a mutation exists which protects from late onset Alzheimer's disease, and that's PLC Gamma 2. And this is now work from Babette Takalon, and who came as a guest scientist from Nico Hilton's lab to my lab. And we wanted to find out what's going on here. And make a long story short, what Marie found was that the protective mutation, which we knocked into a mouse, basically increases the catalytic activity of PLC Gamma 2. And that was, for us, really exciting, because PLC Gamma 2, of course, is part of the TREM2 signaling pathway. So in other words, when you increase the activity here, you will increase all these beneficial effects which are listed here: chemotaxis, phagocytosis, survival and proliferation in the ERK pathway. And in fact, survival and proliferation, as I will show you later, is really, really important and can be pharmaceutically affected by stimulating the TREM2 signaling pathway. So all that points to a protective function of TREM2 in mouse models. But what about humans? Can we get some, at least indirect evidence that TREM2 in humans would also have protective functions? And what I show you now is work from Mark Suarez, who was a postdoc in my lab a couple of years ago, and then replaced by Estrella Sabri, and I will show you data from both of them. So Mark originally started out by simply measuring soluble TREM2 in the CSF of DZNE patients. So these are patients from the dominantly inherited Alzheimer's disease network, where we collect patients with APP, PS1, and PS2 mutations worldwide. He looked cross-sectionally, and the data we have published quite a long time ago. But today I want to show you some brand new data, which are basically impressive and coming out in Lancet Neurology now. And that's a longitudinal observational study, which Estrella carried out in my lab. And we obtained longitudinal CSF samples from 155 mutation carriers and 93 non-carriers. The mutation carriers, again, it's PS1, PS2 mutations, and APP mutations. And what Estrella found, first of all, she developed a new ELISA, which is much more sensitive than our previous one, and has the advantage that it only detects the ADAM10, ADAM17 cleaved TREM2, and not the spliced, the alternatively spliced variants, which also produces TREM2. And we measure soluble TREM2 as a surrogate for TREM2 signaling, because soluble TREM2 can only be created on the cell surface by ADAM10 or 17, and only after the full-length protein arrived on the cell surface. That's the place where it becomes signaling active. So the amount of soluble TREM2, to our opinion, corresponds to the original amount of TREM2 signaling on the cell surface. Now, by applying this new ELISA, Estrella found that soluble TREM2 increases incredibly early, already 21 years before the estimated year of onset in these patients. So basically before the clinician sees any drop in the clinical symptoms. So that's super early. That would be the earliest response right after amyloid. So the earliest biomarker response which we can see whatsoever, way before anything else changes. And what was also very interesting is that a lower baseline CSF Aβ42 levels, which of course represents as less Aβ42 you have in the CSF, as more Aβ you have aggregated in the brain. And this predicts a higher rate of subsequent TREM2 increase. So already a tiny bit of drop of CSF Aβ42 already results in an increase of soluble TREM2. In other words, the microglia start to respond to early seeds deposited in the brain. And they're extremely early because at the same time, when you look at cortical pipette uptake, so which measures amyloid plaques basically, this method is not sensitive enough at this time to measure anything. So this must be a very, very, very early response, and that's reflected, of course, by minus 21 years here. And that fits very well with work Samira did in my lab as a PhD student. And what she did is she used Matthias Jucker's paradigm for seeding. And what she found was that the seeding is TREM2 dependent. So the microglia, if they can be activated normally, they recognize the little seeds, which you see here as little red dots, basically, and they start to eat them up and remove them as soon as they are deposited. When you have a TREM2 knockout, you see immediately that there are many more of these little deposits, and the size of the deposits also becomes different and increases. So this was already by that time in mice also evidence that the microglial response via TREM2 is a very, very early one in the development of Alzheimer's disease. Now, what else did Estrella found? So she found that a higher longitudinal TREM2 increase is associated with a slower Aβ42 deposition. So it's nothing protective, but only when she looks in pre-symptomatic patients, so before their estimated year of onset. And this had clinical consequences. So in the when the pre-symptomatic patients had higher TREM2 increases, their peculiars showed less shrinkage. And this again resulted in a protection of the cognitive decline in the pre-symptomatic patients, actually quite substantially. And this has, of course, now also consequences, to my opinion, for the design of clinical trials. And we may also try to reinterpret, together with Roche, the DZNE clinical trials with the two antibodies they used. And we try to split up the patients which were used in this clinical trial, and those which had originally high levels of TREM2 and those which had low levels of TREM2, and want to see if there are different readouts of the corresponding anti-amyloid antibody and the efficacy of the antibody. Now, of course, such clinical data, of course, you have to confirm in independent cohorts. We couldn't do that in another genetically inherited cohort because DZNE is the only one existing in that size. But what we did is already 2 years ago in ADNI, we could confirm that soluble TREM2 levels, when they are high, to patients with high levels of soluble TREM2, they show a protection of hippocampal shrinkage. There's also those with high levels of TREM2, there seems to be a slower clinical progression, and there's also a better cognitive outcome. And in ADNI, of course, these are sporadic patients, so that also answers the question, is there any difference between sporadic patients and the autosomal dominant patients, which we have obviously in DZNE. So what I want to say here is basically that in principle, we do have data available that TREM expression can have protective functions in humans as well. And that, of course, brings us now to the idea that we may be able to modulate TREM2 function therapeutically. And so the idea is basically to facilitate the switch from a homeostatic microglia to a disease-associated microglia. So how can you do this? So if you look again at processing of TREM2, we believe that the full-length protein is a signaling competent protein, obviously. So if you would inhibit ADAM10, we should increase the amount of full-length TREM2 on the cell surface. And this should, at the end, result in an enhanced activity. And sure enough, this is the ADAM10 inhibitor here. And what we are measuring here is phagocytosis. And phagocytosis is slightly but significantly increased if we inhibit ADAM10. Of course, so that suggests that indeed the full-length protein is performing cell autonomous signaling, whereas the cleavage then basically stops it. Now, of course, nobody would use an ADAM10 inhibitor to treat Alzheimer's disease. I mean, alpha-secretase is anti-amyloidogenic, and of course we know that if we inhibit secretases, we have multiple side effects because the ADAMs and other proteases, of course, have many other substrates. So we need something much more selective. So that was originally just a proof of principle experiment. So how can we be really selective to block cleavage only of TREM2? And the way how we did this was we wanted to generate antibodies, which basically bind at or close the cleavage site, and therefore sterically inhibit the ADAM proteases to access their substrates. And this is work which was carried out by a very gifted postdoc in my lab by Kai Schlepko, together with people of Denali. And I should mention that the collaboration with Denali is an extremely fruitful collaboration. We were very happy that we have these fantastic colleagues in San Francisco. So what we basically did is we we screened antibodies, and our lead antibody, which indeed bound to the stalk region here, it's called 49. And that antibody binds only 12 amino acids, so this is the epitope here, only 12 amino acids N-terminal of the cleavage site, which Kai has determined previously. And that is, of course, closely enough for a bulky, huge antibody to block access now of ADAM10 and ADAM17. And this indeed works and has striking consequences. So what Kai could show was that this antibody inhibits shedding with a very striking EC50 of 3.7 nanomolar. In the same moment, it increases phospho-Syk signaling. So the TREM2 signaling is activated again with a very striking EC50. Importantly, the antibody increases survival and proliferation of microglia and macrophages. And that's something really important, and which turns out to be very similar actually for all therapeutic antibodies which have been identified so far and published so far. All of them show exactly the same behavior. Now, our antibody also drives myelin uptake and amyloid uptake, so it drives basically phagocytosis. So with such an antibody, now you can go in vivo and check what happens to amyloid pathology. So we did a very short experiment here in an APP knock-in mouse model, so which had not too much of fibrillar amyloid, because that may not be removed by microglia at all. And we treated them basically for 10 days with 50 milligrams per kg of the antibody. You have to go very high because obviously you have to get the antibody across the blood-brain barrier. And this had profound effects on the amyloid pathology. So what you can see is that antibody treatment didn't affect the core, so the center of the amyloid plaques, but it seemed to allow the removal of the halo of these amyloid plaques. So the antibody indeed stimulates microglial functions on microglia which surround amyloid plaques and apparently try to remove them. And I just want to say again that a couple of companies like Electro and Amgen, they also generated antibodies, TREM2 agonistic antibodies. And all these antibodies, including our 49 antibody, they have very similar properties, and that cannot be by chance. They were all screened stochastically, basically. So, and we all found antibodies which bind to the stalk region. these antibodies inhibit shedding because they bind close to the cleavage site. And in the same moment, these antibodies also cross-link TREM2, and this also stimulates, of course, TREM2 signaling. So this is a dual activity or action pathway which these antibodies are stimulating. And as I mentioned already, the common denominator of these antibodies is that they drive survival and proliferation of microglia cells. So that may be a common pathway, and I would believe that all these antibodies will maybe show similar effects when we go into the clinic. And actually, Alector already checked in a phase 1b study their own antibody and could show that at least there were not tremendous side effects so far. But talking about side effects, there may be a huge problem, and I worried about this a lot. And one of the problems I actually forgot to mention, we never talked about soluble TREM2. So if we reduce soluble TREM2 cleavage, and we have no idea what soluble TREM2 is doing. So if soluble TREM2 has a protective effect as well, we may consider that, and we have to find out basically what soluble TREM2 is doing before we go into patients. But now another thing which concerned me a lot was a finding which is well known in the field. So when you look in another disease, in FTD, with a progranulin loss of function, microglia are hyperactivated. So that's the case in humans, where you can detect this with a TSP PET imaging, but that's also the case in mice. And I was, of course, worried a lot that if we use 49, that we basically overactivate microglia cells. They end up in a hyperactivated state, and they cannot be brought back. So it's a one-way road where we go from homeostatic to superactivated. And the superactivated, we all believed that superactivated microglia may be detrimental and probably at the very end cause even damage. So there was really something which really, really worried me. So we wanted to answer, first of all, a very important question. And that is, so microglia, obviously, many people have shown that can go from homeostatic state in a TREM2-dependent manner to a disease-associated state. But what about this way? What about going back? This would be very important because if we overdose a patient, let's say with 49 antibodies, can we take out the antibody, just don't give any new antibody, and then hope that the overactivated microglia switch back to homeostatic microglia? This was, I thought, originally wishful thinking, but it may in fact work. So what I'm going to show you now at the beginning, this is work by Annika, a PhD student who's just about to finish her PhD, together with Anja Kapell, a co-worker in my lab who is around since more than 2 decades now, and again with Denelli. So all of them together got some really, really exciting data. So Denelli developed some antagonistic antibodies, so antibodies which do the opposite of the 49 antibody. So they basically block TREM2 function. I will show you that in a second. And when we look at the binding site of the antibodies, they bind here in the IG fold, far away from the position where the 49 agonistic antibody binds, which binds around this position here, close to the cleavage site. So that already shows you that these binding domains can elicit, when the antibody binds, completely different phenotypic consequences. So these antibodies, both of them called now antibody 1 and antibody 2, they nicely inhibit phospho-Syk signaling in a dose-dependent manner. So they do exactly the opposite of what 49 does. Now we went into macrophages from granulin patients in Germany, which we identified here. And the first thing what was apparent was that these patients upregulate TREM2 as compared to a healthy control, which makes a lot of sense because TREM2 drives the activation of microglia to the disease-associated state. Now, treatment of these macrophages with antibody 1 or 2 nicely decreased mature TREM2. And this can be quantitated. And in the same moment, these antibodies also decrease phospho-Syk signaling. So this is a very nice proof that even in cells derived from human patients with a heterozygous loss of function mutation, indeed, the antibodies are doing what they are supposed to do. So now we continued, and that's work now done by Sophie Robinson, PhD student in the lab, together with Dominique Paquet, who runs a lab which is very much interested in human iPSC-derived brain cells, and with ESA Bioscience, a biotech company where we also collaborate. And the idea was to generate a granulin knockout iPSC line and differentiate them into microglia, and then check what's happening, basically. And Sophie did that together with ESA and Dominic, of course. And what we found was that the granulin knockout cells, they nicely increase mature TREM2. And as a consequence, of course, they also show soluble TREM2 increases. Now, when we treat with antibodies, soluble TREM2 was even further increased. And of course, full-length TREM2 reduced. And in parallel, phospho-Syk signaling was reduced by these antibodies. And the phagocytic activity of these human microglia was dramatically reduced as compared to the control. So, do these cells really switch back? So we checked a couple of genes using the nanostring technology, and what we basically found, the genes which were upregulated and correspond to the DAM signature, like SPP1, CSF1, CCL3, and so on, they were downregulated nicely by these antibodies to their basically original levels. And in contrast, a gene like P2RY12, marker gene for the homeostatic signature, was upregulated by both antibodies back to normal levels. So that shows you very nicely that these antibodies indeed switch back microglia from an activated state to a homeostatic state, which I found was very good news. So then we went on and did the obvious, which I admit is a brutal experiment. So we crossed the granulin knockout with a TREM2 knockout. So we generated double knockout with the aim to calm down the microglia in the granulin knockout mice. And this indeed works again, so very well. So I show you again expression data here. So these are DAM signature genes, and they're at least partially brought back more or less to normal levels in the double knockout. So what that means is that indeed we are able to show that microglia which were activated can be brought back in homeostatic state. So homeostatic microglia to disease-associated microglia is not necessarily a one-way road, which is important for any kind of treatment you're going to do, because at some point you may overdo the treatment and then you have to withdraw the antibody and basically go back to bring the microglia back to a normal level. And in double knockout, we found something else which was quite surprising for us. And that is, if you look at NFL, which is a marker for neurodegeneration in CSF, and compare the granulin knockout to the double knockout, so granulin knockout and TREM2 knockout, you see that in the double knockout, where we bring back the microglia from a hyperactivated state to a homeostatic state, that under these conditions, NFL shoots to the roof. So that means that even hyperactivated microglia, which we all, including myself and everybody in the lab originally, believed they may be detrimental, even those guys have some protective function retained. So I think this is very important for any kind of clinical trials, which of course many companies are planning now. And that's the reason why all these companies are of course working on TREM2 agonistic antibodies. So originally we thought that the therapeutic window of an antibody between who drives homeostatic microglia to, let's say, a disease-associated DAM-like microglia, and that this therapeutic window is supposed to be quite narrow. But this is not necessarily the case. These new data basically indicate that there may be a much wider therapeutic window, which allows, of course, hopefully, a safer treatment. But of course, what I'm showing you here, this is still wild speculation, and I should be very careful with making any predictions for clinical trials. We all know how difficult that is. And I want to mention again that though far, we have not addressed the question of soluble TREM2, and those levels we are definitely affecting. And it certainly has a function similar like soluble ABP, where we are all talking about all the time, but the function is not entirely clear, to say it clear. Okay, so now I want to switch gears in the last few minutes and talk about another gene. And that's a gene called AB3. And it's also, there's also a risk factor found at a serine, which is mutated at position 209 in humans, 2-phenylalanine. And AB3 is also selectively expressed in microglia, and it's involved in F-actin dynamics. So in principle, one could hypothesize that the mutation, or that AB3 itself, is functionally involved in maintaining and allowing the morphological changes microglia are constantly undergoing. They are migrating, so they have to grow out their food processes all the time. And for that, of course, you need a very dynamic F-actin cytoskeleton. And so the mutation can certainly exactly affect that. And this work now was carried out by a very special postdoc in my lab by Mariam, and together with Michael, Willem, and Vitali, who is from DZNE. So again, we collaborated here with DZNE. So I want to start out with some biochemical data which were created by Michael Willem. So he simply looked in Western blots of brain lysates from mice where we either heterozygous or homozygous knocked into the CRISPR technology this mutation here. And you plot it here with an AB3 antibody. And what you see immediately, that in the wild type, you have a very strong band right here. So the upper band is very strong, the lower band is very, very weak. In the heterozygous, this pattern directly turns around. And in the homozygous knock-in mice, we only see the lower band. So this is very suggestive, of course, of a post-translational modification. And to make a long story short, I cannot show you all the original data now. Phosphatase treatment, for example, leads to a switch of this band to the lower band. So this is phosphorylation. And what Michael could also show very nicely is that it's not only this position here, this serine at 212, which is affected in mice, but their neighboring phosphorylation sites as well. And all of them are apparently phosphorylated. And if you have the mutation, all of the phosphorylation of all the neighboring sites is also affected. So there seems to be some kind of a constitutive phosphorylation induced by phosphorylation of the serine at position 212. And importantly, this pattern of lack of phosphorylation, we could also confirm in human microglia derived directly from patients. So here we collaborated with Mikko Hiltunen and his Finnish friends and collected IPs, and they collected cells, basically stem cells, and generated human microglia from AB3 mutation carriers. So we find the same pattern in human patients with the AB3 mutation. So if there's a functional deficit of AB3 caused by the lack of phosphorylation, we would expect, based on their function in F-actin dynamics, that the microglia would change their morphology. And sure enough, the microglia changed the number of branch points in a gene dose dependent manner. So these are heterozygous, these are homozygous, and they also changed their mean volume. So there is definitely a change in the morphology of the microglia cells, driven by the misfunction of AB3, which is itself driven by the lack of phosphorylation. So now Mariam went on, and she wanted to stay as close as possible to the human disease situation. So she focused directly on the heterozygous mice. And that turned out to be a really good idea. So what Mariam found first of all was that there's a reduction of at least selected DAM markers like EBA, CD68, TREM2, and CLEC7A in old mice, in aged mice only. So if you look at younger mice, this is not the case. There seems to be an age-dependent loss of at least some DAM signature markers. And this is also reflected very nicely again by TSPO imaging, which again, of course, Matthias Bandel carried out with his group. And what we found was basically that in young mice at 6 months, there's no difference. So I told you at the beginning that these mice basically can increase wild-type mice, even those expressing or making amyloid plaques, they increase their microglial activity. And this happens in AB3 wild-type and AB3 heterozygous mutants similarly. But when we look after 1 year, you see immediately the difference. So the heterozygous mice show much less activation than the wild-type mice. So that means we have an age-dependent loss of a microglial activation towards a DAM signature. Now, Maria made a very surprising observation. Microglia, even in old mice, still clustered around amyloid plaques. So how can this be? Because we showed before, if you have a TREM2 loss of function, there is basically no clustering of microglia cells anymore. So this is a wild-type mouse, and here's an amyloid plaque, and you see lots of microglia sitting around. And here is a TREM2 knockout. There's basically very little microglia clustering around amyloid plaques. So how can this be that in the AB3 mutant, the microglia still reach the amyloid plaques, but with a DAM signature, which at least in the long run is downregulated? Very strange in the TREM2-dependent pathway. So, and Mariam then looked together with Michael on amyloid, on plaques, but also biochemically, and I'll show you just the plaque stainings. She made another very surprising observation. First of all, the metoxy signal, which labels fibrillar amyloid, was the same in wild-type mice and in AB3 heterozygous mutants. But when Mariam looked for antibodies which recognize A beta like 3552 or 4G8, there was a reduction of the amyloid content. And not only that, take a look at ApoE. ApoE is drastically reduced in the amyloid plaques. It's drastically reduced in the amyloid plaques. And when we look biochemically in formic acid extraction, that's what Michael Willem did, is again in the AB3 mutant, we saw a drastic reduction of ApoE, but not in the soluble fraction. And in the soluble fraction, it's mostly astrocytic produced ApoE, which floats around probably in the interstitial fluid, whereas in the formic acid fraction, of course, we catch amyloid plaque associated ApoE. So the Western blot nicely confirms the immunohistochemistry. Now, we have shown before, this again work from Samira, that under loss of TREM2 function, be it a T66M mutation or a homozygous loss of knockout, there's basically no ApoE in the amyloid plaques. And again, Michael could show here that under these conditions, there is much less in the formic acid in the insoluble fraction, in the plaque-associated fraction of ApoE as compared to the wild type. So what does it mean? Of course, we know from David Holtzman's work and from the work of many others, much earlier even, already like almost 25 years ago, or even longer ago, that ApoE drives A beta aggregation. And ApoE in the amyloid plaque basically serves like the glue of the amyloid plaque and keeps the amyloid plaque together and keeps it compacted. And in fact, it has already been shown by Dave Holtzman and others that if you take out ApoE, plaques start to fall apart. And that's exactly what we found when we looked at the closer looked at the amyloid plaques in these ABI3 heterozygous mice. So this is work carried out by Martina Schifferer, who is running our electron microscopy facility here in Haass, together again with Mariam. And what they found was that these plaques are indeed kind of falling apart. And I even started to call them exploded plaques. And actually the term exploded plaque that originated when a long time ago, Samira and myself, together with our neuropathologist, were looking at TREM2 cases, human TREM2 cases. And I thought that these plaques are looking different. And they look like, to my opinion, like falling apart in little pieces. And this is exactly what we see in the mice here. Now, what's known is when plaques start to fall apart, the soluble amyloid, which is then drifting away, ends up at the vasculature. And this is indeed the case. That's exactly what Mariam found. So there's amyloid ending up at the vasculature, and that happens in APP-PS1 mice. And APP-PS1 mice, now talk to Matthias Ducker about that, normally never show any vascular pathology because their amyloid is driven by a presenilin mutation, the L166P mutation, which drives very, very high ratios of beta 42 to 40. So that stuff aggregates right away, and before it can ever see any kind of vasculature. So this was a very surprising result, and Mariam's further investigating that. And what we also found was that there's damage of the vasculature, and there's also a weakening of the blood-brain barrier. So at the end, what that shows you is a novel pathway how a dysfunction of microglia cells can change the equilibrium of amyloid plaque pathology to vascular pathology. And it shows you that probably a compaction of amyloid plaques, that's discussed now very heavily in the field, of course, that a real compaction of an amyloid plaque may even be, at some stage of the disease, may even be a bit protective because it prevents soluble oligomers, which Dennis Selko, of course, claims that they play a big toxic role in the brain. And that this basically prevents their liberation, so they cannot reach neurons and kill them, and they cannot reach the vasculature and cause vascular problems. And with that, I'm basically at the end. And I just want to go back for a second to the amyloid cascade. So I think we have to revisit it, of course. So first of all, there's soluble amyloid, which I myself and many other, and Todd Goldie and others have shown decades ago to be produced constantly from birth on. So amyloid itself, the soluble amyloid, cannot be at the beginning of the waterfall. It must be way up the river, basically, probably where the spring is already. But what's at the beginning of the river must be amyloid seeds. And these seeds drive plaque formations, and then we can discuss how tangles are being formed and if the tangles cause cell death. Nobody knows. There may be many other things in between. But where are the microglia? And I think the microglia are an integral part of the amyloid cascade. So, and obviously they respond immediately. We have shown that in humans even, they respond immediately to the earliest formation of amyloid seeds. So what I really don't like is if people call the microglial pathology a kind of an alternative pathology, an alternative pathway to amyloid pathology. To my opinion, microglial dysfunction driven by these genes I was talking about, probably many other genes as well, which are selectively expressed in microglia, microglial dysfunction really fits into the amyloid cascade. And quite frankly, we should never forget all the genetic evidence, which we already have since decades for the amyloid cascade. So I think rather than coming up with completely new models where we have things really complicated, going in parallel pathways, multiple pathways, I rather believe that this all should make sense in the cascade, which John originally postulated. And I think our data at least suggest, of course, it's still a bit hypothetical, but they suggest that the microglia cells are an integral part of the amyloid cascade. And needless to say that already Alois Alzheimer's long time ago already found microglia cells, not knowing that they are microglia cells, but he described them very well as sitting around amyloid plaques and around basically every single amyloid plaque. And with that, I'm at the end of my talk, and I want to thank again all the co-workers in my lab. And I had the names mentioned, hopefully, at all the slides correctly. So I really want to thank every single one in the lab, and also the previous lab members who in the meantime do their postdoc in other labs or already started their own career. And I also want to deeply thank the entire Denali team. So we have, as I said, a super close collaboration, basically in all our microglial projects without any exception. And this is one of the best collaborations I have ever had in my life. I also want to thank the ESA team, small biotech company here in Munich. And I want to thank, of course, all the other collaborators. I mentioned Dominique Paquet, who runs the IPC lab basically here in the Institute, in the Institute for Stroke and Dementia Research. I also want to thank Mika Simons, of course, very important, Matthias Brendel, who is running all the PET imaging studies for us. And of course, neuropathology, there's Thomas Arzberger, Regina Federle, of course, and Sabina Tahirovic. And finally, last but not least, I want to thank, of course, the clinicians who are running Diane and Edney. And of course, I also want to thank all the patients and their family who contributed to our research by participating in these cohorts. And actually, Diane, we are actively recruiting here in our institute as well. So many thanks to all of you, and I'm happy to address questions. Thank you.